A very good morning to all of you and a very warm welcome. I especially welcome Professor Tracy Bansal, who is valedictorian lecturer
Recording in
speaker.
progress.
Professor Bansal is a renowned figure in plant biotechnology in India. By training, he is basically a plant physiologist, but he has done pioneering work in transgenics and molecular biology research. Professor Bansal was principal scientist and professor at NRCPB at that time and NIPB now. And then he became director of NBPGR. And in NBPGR, he has led a very big program on wheat genotype screening. And if I am not wrong, sir, that program is recorded in Limca Book of Records. That was a very big challenging program. So we are fortunate that Professor Bansal is here with us for delivering his lecture. I invite sir for his lectures. Yeah.
Well, thank you very much, Dr. Anil Kumar Singh, for this kind words and inviting me here to give you this valedictory talk, the last presentation today of this training workshop, which was organized from July 1st to today, July 12th. And I'm very happy and congratulate the organizers, including the director of this institute, Dr. R.C. Bhattacharya. And I also, of course, congratulate you all that you are the very few ones out of those thousands of applications selected, for this particular training program. And the topic assigned to me, as you can see, is advances in genome editing, challenges and opportunities. So my focus would be mostly on challenges and opportunities. And I'm sure by now, you would all know what about genome editing. So I'm not going to tell you about genome editing. And I would like you to kindly be attentive, having an eye contact with me. And I also welcome those participants who have joined online. I'm sorry I can't have an eye contact with you, but then I would certainly request those who are present here in person to listen to me carefully. This is my request, to have an eye contact. And if anything is not clear, they will ask me. And since this is the last presentation, we have about an hour with us. So initially, 20 minutes I like to spend to interact with you, to learn from you, right? And of course, then I will prepare myself for the presentation and talk to you and give you that lecture. Why I'm doing so, this kind of interactive session, simply because that I have gone through the whole program. And again, I congratulate organizers. This is a wonderful program they lined up for you. Excellent speakers and very good experiments and practicals. So therefore, by now, I'm sure you're quite familiar about the whole technology, its application, etc., etc., right? And I'm going to talk to you about challenges first, then opportunities, obviously. Okay? And also, I know that you're all masters, few of them, mostly PhD students, and across the country, basic sciences, applied sciences, agriculture, product development, and the need for the farmers, and to be self-sufficient, to be having national food security, nutrition security, and also contributing to the global food security, right? So now, without wasting much time, 9:40 and until 10 o'clock, 20 minutes, I would like to now know from you to start with, before my presentation I open up, and from each one of you, right? I will start from there, right? Because what I'm going to ask you, you have to immediately think and tell me. So I'll come back to you again at the last, because you're the first one, you will have least time to think about it, because the question will be the same, which I'll be going to ask all of you. My question is now, and why I'm asking the 12th day of your training program. What's your good name? Arushi. Arushi, according to you, what are the challenges? What are the challenges around genome editing? Whatever challenges you think of utilizing it for crop improvement for which this training program has been organized. So accordingly, what are the challenges?
For CRISPR or
See, I'm asking you, this is my idea to learn how much you've learned during the last 12 days.
okay.
The topic is very clear, it's CRISPR based genome editing only and for crop improvement. We are in an agricultural setup. So my question is, according to you, what do you think are the challenges for using this technology for crop improvement?
So basically,
Immediately you can think of, you can tell me one or two.
due to increasing population, we
I'm
have
asking
to
technical challenges.
technical challenges.
Because you are a user. Okay, I should specify further. As a user, you want to use this technology now for a given crop to improve a particular trait. What are the challenges?
So I think one of the challenge can be the efficiency that is very low. So we have to screen a lot of plants
Okay.
to obtain that one mutation,
you. Sit down.
desired mutation. Thank you.
You got the question now? Should I repeat? No.
No, sir.
So what are the challenges according to you?
So for me, the first challenge is the acceptance.
Huh?
Acceptance of this technique.
No, I'm talking technical challenges, madam.
Okay, technical challenges. So for technical
अभी तक
challenges,
वहाँ नहीं पहुँचे हम लोग.
yes. Okay, sir.
You
So
all
for
understand
me,
Hindi, by the way?
yes.
Okay. So what are those technical challenges? Again, I repeat, technical challenges. As a user, you want to use technology when you go back to improve a particular crop or a particular trait.
So for me, it's a time-taking procedure. And after that, after putting a lot of effort, we are not sure that we are succeeding or not.
Okay, please sit down. Thank you. I don't have to repeat the question, I believe.
No, sir, no, sir.
Thank you. Tell me, please.
Off-target effects will be the
थोड़ा दूर रखो.
off-target
Okay.
will be the major challenges
Anything?
and a rigorous tissue culture process for which we do the transformation.
Thank you. Sit down.
Thank you, sir.
Good morning, sir. For me, major challenge is like
You are from where? No, just basic बोल दीजिए. You? Okay. And you? Okay. Now you please.
I'm from RLBCU Jhansi, sir.
Okay.
For me, major challenge in genome editing can be like designing vectors, sgRNA and all, guide RNA, which we need expertise for that, sir. Like,
Okay. You got that expertise here, of course?
yes, sir. Means-
Wonderful. Please sit down.
Thank you, sir.
You are from where?
So I'm from ICR CSSRI Karnal.
Right.
So for me, the major challenge is like salinity stress.
Those are challenges of the life of the crop. I'm talking technical challenge for developing a crop using genome
So
editing.
to insert any salt tolerance gene in crop like wheat.
How do you insert? Is this a challenge? How it is a challenge? Anyway, sit down, please. Thank you. Quickly, you have to be very smart and quick.
So myself, Yamini from IHL Bangalore.
थोड़ा
Can
दूर
I
रखिए
speak
उसको
like
mic
that?
को. हाँ.
So the challenge would be maybe precision. Precision would be the challenge.
Precision is an advantage of this technology.
So, but like in many ways,
तो
we.
ये target से relate कर रही हैं क्या?
Yes.
Okay, sit down, please.
So I am Munmun Kothari from GB Pan University, Pannagar, and I think the major challenge for me would be the designing of guide RNA, targeting of exact sequences that I would like to edit for the crop.
Wonderful. Thank you. Sit down.
Thank you, sir.
So myself Neelam, I'm from CFTRI Mysore. So the background I come from, I'm nowhere related to this, but I think sequencing the guide RNA and then incorporating it into the crop itself is a big challenge.
Okay, thank you.
Good morning, sir. Myself Sindhu. I'm from IRI New Delhi. Major challenges is identifying the gene which are having a negative regulation is one of the main
Sorry, say it
disadvantage.
again. Sorry.
Sir, since in genome editing in CRISPR, we are mainly targeting for gene knockout. So identifying a gene which are having a negative regulator so that we can going for a knockout is a challenge.
Wonderful. You are from where?
IRI New Delhi, sir, genetics division.
I?
IRI genetics division.
Oh, IRI. Okay. You're right. Thank you.
Good morning, sir. My name is Adira. I am from IRI only. I think
identifying the target gene and deciding the vectors,
Are
everything.
you good friends? Eh? She's already mentioned that. Okay,
No.
thank you. Sit down, please. Yeah. Any other than target gene? Any other from you? No.
No.
Okay, thank you. You.
Good morning, sir. Myself Ayushi Singh. I am from RBS College, Agra. For me, it's like identifying the gene that is causing a problem and then sequencing it further. So identifying that gene can be a major issue in the beginning.
Wonderful. Thank you.
Good morning, sir. My name is Sejal Batra and I am from ISR Lucknow. And according to me, the major challenge will be off-type effects and genotype dependent transforming principles for every.
You're from
ISR Lucknow.
Lucknow.
Yes, sir.
Sure, you will certainly be able to do genome editing in sugarcane, right?
Sir, it is a polyploidy and it
But
is tough.
It's not difficult. Already people have done it. Talk to Anil, Dr. Anil Kumar. He will help you.
Yes, sir.
Okay, next.
Good morning, sir. Myself, Priyam Saini from Amity University. I think major challenge is to transformation of that gene in the plant and the designing the guide.
You got some idea now from the previous speakers.
No, no, sir. It's like
Wonderful. No,
major
but this
thing
is
now.
good. Actually, that was the whole idea.
Actually, we are trying to do that, but
Which crop?
rice is that.
Okay. All right, great. Next, please.
Good morning, sir. I'm Anuradha from PJTSU.
From?
PJTSU.
Okay,
sir.
Hyderabad.
Yes, sir.
Huh.
I think availability of genome sequence for some crops like millets, I think is one.
Okay.
of the and also final results, like we may get positive results, but that expression may not be sufficient. And also the
Expression may not be
Not be-
sufficient of what?
of the edited gene, like
Think over it again. What you're talking. Think over it again. You undergone a training.
Yes, sir.
Expression of a gene you're targeting.
Yes, sir. Sometimes I think final results, sir.
Expression
Like,
low or high, or what do you want? The expression.
that is unpredictable.
Unpredictable.
For,
Okay, thank you. Sit down, please. Next.
Good morning, sir. I am
morning.
Khushbu from CSIR IHPT Palampur. So, according to me, designing guide RNA is major challenges. And apart from that, standardization of transformation protocol is also a challenges.
Okay, thank you.
Good morning, sir. My name is Parul. I'm from Himachal, and according to me, the major challenge is selection of gene, and sometimes we need, as a student, technical expertise is necessary in genome editing, I guess.
Okay, thank you. Sure.
Good morning, sir. Myself Harshita Singh from IFGTB Coimbatore. And according to me, I guess selecting a gene for a particular trait is and standardizing the protocols for that particular crop will
Okay,
be.
talk to Anshul. Dr. Anshul will just come here. He will help you more. Next.
Good morning, sir. Myself Nisha, and I am from Thapar, Patiala. And according
from
to me,
Thapar, Patiala?
yes, sir. And according to me, it could be optimization of regeneration and transformation protocols for the crop.
Which crop you are working on?
On potato, sir.
Okay, thank you. I was there in the board of Thapar Institute, by the way.
Good morning, sir. Myself Pooja Dev from Assam Agriculture University, Jorhat. I think the major problem of genome editing is for complex traits which are controlled by multiple genes. So we need precise editing of multiple loci.
You're from?
Assam.
Assam, which
Agriculture
university?
University.
Assam Agriculture University.
Yes.
Okay, thank you.
Thank you, sir.
From the back, शुरू कर दीजिए वहीं से. Yeah, please.
Hello, sir. Good morning, sir. Sir, I am Kailash Patel from SKRU Bikaner. Sir, as a pathologist, the major challenge is to identify the gene from the whatever the biocontrol agent to improve the efficacy.
Okay, thank you.
Good morning, sir. I am from GKVK Bangalore. My name is Nimish Bhushan, and the major challenge in CRISPR-Cas9 is that it requires well-characterized genes, which is amenable for editing. And apart from it, the standard tissue culture protocol should be there, and which is not available in many of the crops right now. And apart from it, the off-target effects is more in case of
Okay,
CRISPR-Cas9.
you're from where?
GKVK Bangalore.
थोड़ा ध्यान से धीरे बोलो यार.
GKVK Bangalore.
Bangalore.
USB.
Okay.
Sir, Mohit Agarwal from Sukas, Jammu. And the challenge would be the off-target effect of guide RNA.
Okay, thank you.
Good morning, sir. I am Akshay Tavle from ICER Tirupati. So, one of the major challenges that I can think of is creating the overexpression lines with the gene editing techniques, because the knock-in could be the having the very low efficiency when we are talking about the HDR efficiency, creating external sequence into the cell along with the CRISPR sequences.
Okay, अभी ट्रेन थोड़ी लेट है, announcement हो रही है.
Good morning, sir. I am Rushikesh Mane. I am come from Dr. RPCU Pusa, Bihar. So, sir, everyone highlighted the major technical issues like target gene, designing RNA. But, sir, I want to tell one thing. Sir, after editing the complete cassette through cassette, so screening that edited one from the population, whatever we got, after means that cell where
Okay.
our genome editing.
All right.
This is the one technical, sir.
Thank you.
Yes,
Sure.
sir. Thank you, sir.
Next.
Good morning, sir. My name is Sajit Kumar from Kerala Agriculture University. Sir, in my case, I am working in genome editing in banana. For me, tissue culture part is generation of somatic embryo and the regeneration of the transformed
Okay.
is difficult.
Thank you.
Thank you, sir.
Next.
Hello, sir. My name is Shobik Chandogi. I am from BCKB, West Bengal. For me, sir, I think offside mutation will be the problem for genome editing.
Okay.
Thank you, sir.
Good morning, sir. Myself, Sana Singh from CAU Imphal. My major, like, I'm thinking like most of the plant or crop have not been sequenced, so the major problem will be sequencing problem. Thank you, sir.
Okay.
Good morning, sir. I'm Chinmay from US Bangalore. So for me, the gene finding is the major problem, and later on, if we also design the SGR and all, incorporating into the plant is and making the protocols for regeneration is a major.
Okay.
Thank you.
Next, please.
Hello, sir. My name is Ashish Chovatia from Junagadh Agriculture University. My major problem is groundnut tissue culture protocol and transformation.
Wonderful. Anyone else left? Are you all 35 in number, or anyone who is absent or who came just late? Anyone who has not spoken to me about it yet? Who came late? It's okay. You can tell now. I'm asking everybody any kind of a technical challenge. What you think according to you should be?
Good morning, sir. My name is Muskan. I'm from Delhi University. So the major challenges for me is the off targets. These are the
Okay. You want to say something still? Or you mentioned what was your problem? You said challenge? Okay. Well, thank you very much. Are you all still 35 here? In number? Okay. And you can hear me well? Yes, somebody online?
Sir, actually, I am also a trainee, sir. Due to some issue, I couldn't come there. I'm late.
Okay, what's your good name?
I'm Badrika Himatindu, sir. I'm from Andhra Pradesh.
Okay, you're online, right?
Yes, sir.
So what's your
Because I
question?
couldn't come, I joined online.
No, that's fine. But you have any question?
Not question, sir, but I would like to add on like
Yes?
specificity is the problem in genome editing, no, sir. Specificity of guide RNA.
Okay. All right. Thank you. Thank you,
Yes, sir.
Madam Hima Bindu. Thank you. And thank you, friends. Thank you very much. You have done half my job already. That was my first half of my lecture. Right? You know about the challenges now, but still I will go over with you. Okay? Some of you were very right. Some of you were still thinking. And the whole idea by now, today, on the 12th day, the last day of the training program, is that you should be very clear what are the challenges. As a worker, as a technical expert now, you know, that you have to deal with a particular crop, a particular trait, because here we're talking about use of this technology for functional genomics, mostly for also crop improvement. So what I will do now, I will quickly go through some of those slides in my presentation, next about 30-35 minutes, and still we have time. I'm sure you will be welcome to ask me questions. Okay. Next slide is, these are the technical challenges. Okay. Some of you have covered it already. I'm very happy about it. But those who are not clear may kindly note it down now. What are those challenges? First is identification of the target gene. Without target gene, you cannot move further. Functional transformation protocol, you said in a crop, but I would like to clarify, in a particular crop of that particular elite genotype or elite variety. That's important. Please note down. So that particular genotype. Okay. Third, then developing effective method to deliver genome editing reagents more efficiently into plant cells. That most of the plant cells are able to have that kind of editing which we require, and that also depends on the plant species and the tissue type. Plant species means the crop. Within a crop, the variety has to be an elite variety, and tissue could be where you can regenerate the whole plant. So it depends. So these are the technical problems. Increasing the efficiency of editing process to ensure that these genetic changes, they are made reliably and consistently in higher percentage of cells. Means whenever either you bombard or you do agrobacterium or do your protoplast, this you have all learned it. So the efficiency should be high, that large percentage of these cells are transformed and the DNA is edited. That's what is the increasing the efficiency. Now when we talk of increasing the efficiency, I should clear that point right now here. You know about SDN1, you know about SDN2, someone talked about it, that they want to overexpress it using HDR, fine. Number 3 is that they want to overexpress even the whole gene, for example, using SDN3. But now compared to SDN1, SDN2 and SDN3, where is the editing efficiency most highest? SDN1 compared to SDN2 and SDN3. SDN3 hardly you will find globally people are using at the moment, because you have a classical conventional GM crop technology available. But this is of course an advantage, no doubt, when you use genome editing or using SDN3 to overexpress a particular entire gene from any other organism, including from the same plant species, suppose you want, or related plant species or anything. But efficiency is highest. People have attempted so far mostly SDN1. That means knocking out the gene function. Okay. And then SDN2 is very little, because efficiency is very low. You have to use HDR, and you have to also use HDR, of course, even in SDN3, their efficiencies are low as of now. Then editing complex traits that involve multiple genes. Only one person, I think from where, Delhi University or somewhere, she mentioned about it, from Assam Agriculture University, sorry. That's very important again, because some of the quantitative traits you want to edit, yield, for example, per se. Of course, there are now subcomponents of yield, and genes have been identified, particularly transcription factor encoding genes and otherwise, where single gene can still help, but still you need multiple genes. In fact, even to increase the regeneration efficiency, as one of the problems you all mentioned about it, there could be another gene editing, when simultaneously it will increase the efficiency of regeneration and also will edit the trait of interest which you want to. So you can read more about it. It's called combo kind of gene editing. Then an efficient DNA free, this is altogether a different ball game for particularly vegetatively propagated crops, wherein you don't allow your CRISPR reagents to be stably integrated with the nuclear genome. They'll be transiently expressed, or they will be the protein complex itself, which will be in fact kind of infiltrated using protoplast or PEG mediated transformation. There are many methods for which we can try to use it, and that will allow you not to worry later about its stable integration into the genome, so that you don't have to worry about it throwing out later, which is an essential part. In kind of annual crops, it's possible, but in case of sexually vegetative crops, it's not possible, through sexual hybridization, as we normally do with the selfing or crossing of the T0, gene editing crop which you will have developed. Is it clear? So you must therefore go over it individually, okay, in your crop, your trait of interest, and whatever you want to do. This is your immediate exercise, actually speaking, after you go from here. These are the technical challenges. Now, there are other challenges as well. And what are those other challenges? Off-target effects. Many of you actually talked about off-targets. But why I put it in other challenges? These are the main challenges which you have to anyway overcome always. Many of you mentioned about this. But these I have listed them. Other challenges are of course very important. But off-target effects, I've kept it to one intentionally because I knew many of you would be talking about it. And you mentioned. But then I must tell you, friends, we can talk about it later. Time will not permit to give you the details. You should not worry about off-targets in plants. Okay? For this, you should read about chemical mutagenesis or mutagenesis of crops by irradiation, where more than 3,000 varieties already developed, being consumed around the world, including in India. We don't know how many off-target mutations would have certainly occurred, to achieve that particular trait which we wanted to, out of thousands of those mutated lines. So here also, as long as, remember one thing, if you're listening to me carefully, as long as your phenotype, your expression of the trait which you're looking for is fine, plants are all fine, you don't have to worry about genotypically about the off-target. Clear? In plants. We are talking of plants. Next, of course, regulatory compliance is very important. We will talk about it. You should know about that. It's another challenge. Integration of genome editing with breeding programs. This is extremely important. Please remember, integrating the program of genome editing with breeding programs, because breeders will only tell you which variety of a particular crop you should be working on for which trait. You are not breeders, right? Neither you would like to turn yourself to be breeders after your PhD doing in a particular subject, you're talking about it. But you must integrate your whole concept and idea and collaborate with of a particular crop. Breeders are mostly of a particular specific crop. You must talk to them. You must understand. Okay? So that means your planning of your experiment must take a little longer, 3 to 6 months. But so identification of those target genes, variety, etc., etc. I don't want to go repeat details. We can discuss about it. But I'm sure you got the message. That is very important. Then public acceptance, that you leave it to us. I mean, government of India is taking care of it very well. That way, particularly I'm talking of genome editing. You got it? So this is all you must know. Now quickly going through step by step again. This is a kind of a summary. I'm helping you revise your thing you would have already learned. And if you're not learned, you can raise your hand so that we can improve, and I can request Dr. Anil Kumar Singh in the next training they can give more emphasis on that part. Now, for example, to start with what? Define the trait of interest, number 1, in a particular crop, of course. Then utilize genetic and genomic resources, available transcriptome data, and what is the genetic basis of the trait you must know. These are all available in literature. Okay? And employ bioinformatic tools to identify the candidate genes based on sequencing, homology, functional annotation, etc. is all there. So what the extra message you have to get from here? You are lab workers. You're working on bench. You're doing a PhD. You will continue working in your bench, but then you must learn bioinformatics. So what you should request the organizers, sir, kindly organize one course specifically on bioinformatics. Give us all information how to identify the target gene based on the transcriptome data, based on whatever the information is available in different softwares, and then of course accordingly you design your sgRNA. I'm coming to that. Literature review to identify previously characterized genes. People have characterized so many genes already for the trait you are looking for. So go into the literature. This is very important. Then functional relevance of that candidate gene through expression studies, etc. For example, RNA sequencing under different conditions and relevant conditions, you have to study it yourself. People would have studied it also. So you have to go through all this before you really speaking, come to the conclusion, this is the gene, this is the trait for this particular crop, for this particular, environmental conditions or different conditions. Then this is very important in your own lab. Then you would like to, having done all this, then you will do functional validation. Functional validation, you would like to know, for example, in a model system, Arabidopsis, and by the way, when I'm talking about bioinformatics, Dr. Anil Kumar Singh, fortunately by himself, is an expert in that area. He would have given you more lessons, but again, I think a specific training is required, right? In 12 days, you cannot learn in each and everything. So that training itself should take you about 7 to 10 days on all this. And functional validation, you would have done it here, not practically possible in 12 days, but you must do it in your own lab. Once you, okay, this is the gene. Now you want to doubly sure, using a Arabidopsis system as a model, or even tobacco, or whatever works with you, you would like to functionally validate, yes, this is the gene, right? Then you do and design guide RNA. Right? So you can see so many steps before you go to that. Then delivery and transformation. We have talked about it already. And when you design this guide RNA here, someone of you mentioned about the challenge is to have that proper genomic information in a particular crop. They were talking about millets. Someone gave an example. Though millets, there are sequences available. But you can always go to the next, related species where the sequences are available. And some of them you find them conserved cross planta. So one can take the advantage of that. But that's important, how to design this. Followed by delivery and transformation. So once you develop that construct, delivery and transformation of those into plant cells. Delivery, as I mentioned before, could be biological gun, could be, mostly it is agrobacterial mediated transformation, but you all must understand about the vectors, how to clone your construct into that particular vector, what is the design of that vector, etc., etc. You must know on what kind of promoter sequence you must use it, and all this. And here, by the way, we are not talking of overexpression when we're talking of particularly knocking out of a gene, using genome editing. Someone mentioned about it. We would like to express the gene. That is only SDN3, or developing a transgenic plant using any other kind of a gene which is related to a particular trait, could be from any source. Then finally, you have developed, for example, during these delivery and transformation methods, you got genome edited plants regenerated. Then you have to characterize those. And someone said sequencing is the problem. Sequencing can be outsourced, or you can do in PCR. You would have learned all this, I'm sure, already here. How to characterize, how to validate that your plant particular sequence is edited. There are ways to do that. There are methods. You would have gone through it. Then finally, you evaluate them for that phenotypic changes for the trait by growing them in the pots under confined conditions as per the regulatory guidelines we have in our Indian system. Then you finally go to the field and you test them in the field. So this is the whole training course actually should have covered. I'm sure it has covered already. If not, bioinformatics, for example, is very important based on the available genomic sequences around the world in a particular crop or for a particular trait. That's really speaking the starting point. But come back to that point is defining that trait of your interest is very important. Not what you need. For that, you have to discuss with breeders. And what kind of trait will that be? Who will use that trait? Breeders will use it? Or who will use that improved trait in a particular crop? For whose benefit will that be? For the farmers. Remember that. Not that you do anything, of course, for your own knowledge, generation of new knowledge, ideas, original thinking, and all, you can do that as a science. Nobody stops you, depending upon the mandate of your institution you're coming from or your own interest. Do some basic studies, no doubt about it. Okay, even functional analysis, knocking out the gene. I mentioned before, here, for example, functional validation, you can even do it using CRISPR-Cas technology. Knock it out and see whether the plant has what kind of phenotype. Is that right? Then I come to opportunities. Okay, what is the burning opportunity you have before us today anywhere is development of crops with inbuilt climate resilience in all major crops. Climate is changing very fast. You would have seen the temperature in Delhi touching almost 50. We never saw it before. And that's the story with rest of the world, glaciers melting. I don't want to give a whole lecture on it, but still I'll show you some slides. So that's a very important trait for all the crops, but particularly when we talk of India, the opportunity is, can we become self-sufficient in oilseeds and pulses? You know about oilseeds? Some of you are not from agricultural institutions, I can understand it, but as a general knowledge, they must know, as an Indian, that we are importing oils, okay, large quantity, costing us about 1,50,000 crores of rupees annually. It's too much of money we are spending. So can we reduce that import? Can we produce more? And similarly, pulses, protein source for us. Then utilize, now important is, if I go back, I didn't mention to you about here, when you want to, identify this gene, candidate genes, you have to look for natural variation which is existing in nature. I will talk to you about that. And that is actually speaking, here is available in gene bank accessions. You have to, I'll send a set of those slides. It will be shared with you all through Dr. Anil Singh, and you will be able to see. And also genomic genetic resources contain these wild crops, wild species, and neo-domestication. I'll give you some examples. Then undertake focused research on functional genomics. I mentioned before, using all this naturally occurring genetic variation in these different accessions, okay, for a particular trait, you will be able to identify, narrow down, and then go for functional genomics, using, CRISPR again. And then identify and validate. This is another important aspect, which anyone who is not coming from agricultural background directly, but you want to use the biodiversity in bacterial species mostly, then you can even look for, other than Cas9, a kind of a different, protein which can, create indels in plants, or can create that double-stranded break. Not only you have to always use CRISPR. I mean, that's a kind of your own research when you can add value using bacterial species, which are plenty available in our country. Now, quickly go through it. I've got only about 20 minutes left, maybe 15 minutes I have to cover. I will go a little fast. Are you able to understand what I'm talking about, please? So I'm just giving you the urgency of climate change. That's what I started with as an opportunity. Climate change, we want to contain our temperature increase, which is happening anyway, but we want to contain in this 21st century and up to 1.5 increase only. 1.5 compared to pre-industrial era. But people are talking we might even contain within 2 is also fine. Number of weather events, whether drought, floods, and all you can see is increasing. There's so many here affecting the crop production. And also food system itself is contributing. We're using 80% of fresh water, 70% of fresh water. 1/3 of greenhouse gas emissions from our agriculture, food systems. So we need to curtail, these have to be cut down by half in the coming decade. So if you want to work on climate change, you have to work on reducing greenhouse gas emissions mostly. Okay, and this is what the World Bank actually talks about it: increasing productivity, enhancing the resilience of our crops, and reducing emissions is all very important. So finally, therefore, what is the relevance of CRISPR? CRISPR can be useful in increasing yield and land use efficiency, to increasing the productivity per unit of land. Yield means you have to increase the yield continuously anyway. That remains your major trait, increasing the yield per unit land area. Okay, and then making plants resistant to those which are all climate related changes or traits. Whether you're talking of insect pests or drought or high temperature particularly. Nutrition has to be combined with it, and then helping farmers finally with those varieties so that they are adapted, their conditions, they all get adapted, with this availability of these genotypes which are improved for climate change. You got it? Then for the climate change, what we have to do, again, I emphasize reducing greenhouse gases, right? From emissions from, which is possible. Then minimizing the farming inputs. You have to increase the efficiency of utilization of fertilizers and pesticides. So on one hand, you're reducing greenhouse gas emissions. On the other hand, you're trying to increase the utilization efficiency of natural resources, be it water or land, by producing more, okay? Or your, nutrients like fertilizers, right? So that farmers have to buy less amount of these resources, and then you increase their efficiency to utilize more, and finally you have to increase more carbon content in the soil. So actually, you know her? She's the Nobel laureate. And this is her message I want you to read carefully, that we should use genome editing to build climate resilience. She's giving emphasis on climate resilience. I'm in the climate resilience area still. And environmental sustainability is very important, that we don't want to damage environment anymore, which agriculture is anyway doing it, but we want to reduce the environmental footprint of agriculture by developing those now genotypes and crops with improved traits which are even climate resilient. So technology allows us. That's what she's saying. And also at the same time, we must preserve and use our agrobiodiversity, rich agrobiodiversity. I mentioned to you about rich agrobiodiversity for either looking for more Cas9 protein genes, okay, or for looking for those target genes for particular traits. The target genes people have already studied, but you may like to go and find out, confirm it for yourself about that natural variation. Is available. In fact, I'll give you some examples, right? And I'm just showing you that I have met her. And I was very pleased actually that she invited, and Dr. Vishwanath and myself had an opportunity in January this year first. We had gone there, and we spent some good time. Because of our own interest in this area, she was, I believe, happy to know that we are trying to do something that way big in India as well. Okay, then integration. Again, this is an emphasis here to use those natural occurring genetic variation. Okay. which is available. Do you know about plant genetic resources? Enough here in the hall? Anyone who knows the best can raise her or his hand. Do you know we have a national gene bank here in India? NBPGR तो ठीक है, नेशनल जीन बैंक है क्या हमारे यहाँ, है ना? What does it contain? Germplasm resources, different accessions, different lines, different kind of material of which is genetically variable naturally. Okay, even including modern varieties or land races or farmers varieties, all conserved. So they have those genes for the traits you're looking for. So that's the whole message I'm trying to give here, and allelic diversity. And here you can see here why these genetic resources become suddenly so important with use of genome editing technology. Because see, this is the modern kind of, this is the kind of a pathway through which these modern varieties have been either selected in early domesticates or later developed using plant breeding technologies. And these are the wild species which are conserved in the National Gene Bank or in their natural habitat as well. You can see different kinds of genes. There are so many. They got reduced with early domestication, okay? And they got further reduced, you can see here, using plant breeding methods, right? Because we were focusing on increasing yield mostly, which is true, which is important. But then also if I wanted to transfer a gene here, from here to here, it will take 10 to 20 years. But genome editing allows to do that kind of job, in 2 to 4 years itself. Genome editing allows that you can, even this wild species, for example, of tomato, this is a wild species. Through genome editing, some of those 5 to 6 genes, target genes, you can develop into a cultivated type. This is an example, okay? And here you can read this that allows you to retain those traits which are important for climate resilience, okay, or survival under harsh environmental conditions, because that's how these wild species have been able to survive. They've evolved themselves. They've got those genes which our modern varieties do not have. So two things here: either you take a gene from these wild species and put it into these through conventional breeding or whatever the method, that takes a lot of time. But that's still possible through genome editing. I'll give you some example. Or you do the genome editing of this wild species itself. We call it as neo-domestication, to save that process, as has been done in case of tomato and also in case of rice it has been done. In rice also it is done in 2 to 4 years, wild type to cultivated rice. And this is the other way, that you can take genes from these genetic resources, those target genes. How will you know what target gene for a particular trait, where are they? So this is the source, actually speaking. You have to characterize them. Then functional genomics using some of these approaches. Okay, identify the natural genetic variation, and then you can install that natural genetic variation. Could be a new allele altogether. You found it, though it's already there in a modern cultivar, a gene, but you got a different allele. Means somewhere, either in the coding sequence or in the promoter sequence, some different genes are sequences or bases are different, right? So you can do that now, kind of genome editing using CRISPR technology, and finally you develop. And that you can do it in locally adapted cultivars. Again, I mentioned about earlier high yielding cultivars, very important, some of the major crops. But some of the crops, farmers prefer only locally adapted cultivars, which are very good in nutrition, for example, but not high yielding. So you can even focus those, using genomic technology based on the available information, in these genetic resources. This is one example of EPFL10. This is epidermis, pattern forming like, gene. If you mutate this, knock it out using genome editing, SDN1, the number of stomata, idea is to conserve water, right? Because plants transpire too much of water. Why do they do it? Because the stomata only through, they take in CO2, which is very important. But can you have stomatal density optimized that they, of course, will lose the water? It's important because we want to keep the stomata open because the CO2 to come in. So can you conserve, at the same time allow enough photosynthesis? That's not curtailed. So here, using, two different genes there. One of the genes, particularly this, you can see here, stomatal density has come down, but this is the wild type. It is very high, but if it has come down here, it will conserve moisture without any effect on photosynthesis. This is very important, and this is work from IGI, by the way, Institute of Dr. Jennifer Doudna, and this is in one of the leaves, leaf 5, and this is in the flag leaf in case of rice, the same kind of good result. This is a natural variation in African rice. African rice is this, Oryza globirima, Oryza glabirima, right? And this has got a allele which provides thermotolerance. And Oryza stiva is not having this gene. So this is of course not genome editing, they've overexpressed it. I'm just giving an example that there is a natural genetic variation. Here in case of rice, in this particular species, this is highly thermotolerant, whereas Oryza stiva is not. So if they overexpress this gene from this particular wild species, into Oryza stiva, you can see plants become quite tolerant to high temperature as well. So you can look for more natural genetic variation, okay, and you will find some, kind of positive regulators, or, you can try to see what best use of you can make. Similarly, this is from barley. We want to reduce the methane emission from rice, paddy fields, right? Paddy fields are flooded. Roots have some exudates. There's a microbial activity. Methogenesis, they call it. As a result, the methane gas will be emitted, mostly from methane, from paddy fields, not from other crops as much, right? But here they have identified this particular transcription factor, which will allow, but again this is through overexpression by the way, but which will allow the translocation of photosynthase to more to the shoots, less to the roots. Okay, because the photosynthase which allow those exudates, it seems, to release into the soil. Okay, and there the microbial activity will be there, and there will be methogenesis, more methane will be emitted. So if you translocate more of this photosynthase to the shoot, that doesn't happen, as has been shown in this paper published in Nature. And this you can see here. This is in Nippon bear, I believe, wild type, and this has been reduced drastically to this level, in this particular stage, and here you can see here further reduced drastically. These are some of the examples. So genome editing, just to again another 5 minutes, I believe, still I didn't realize I'm going to take too long. Normally I finish my talk well in time, but I've got a few more slides to show with you. You're okay so far? You understand what I'm saying? Okay, now here actually now this is a happy moment for us. We achieved something actually speaking with regard to regulation. That was in 2022. So we immediately came up with this kind of a paper with Dr. Vishwanath and others, Dr. Mola, both of them have given lectures here, I know, that what are those traits, that exercise we have already done for you, by the way, not for all crops, some of the prioritized crops, for 10 to 12 crops. But what we find here, this is the category of crop, for example, pulses. These are 3 different pulses. What are the traits? We talk to the breeders. What are the traits? What are the target genes? And whether you should use SDN1, 2 or what? Simple, right? And to our surprise, talking to breeders so important that we got to know about the traits, okay, because they know what the farmers need. But then we did an exercise in-house, searching literature, etc. I mentioned before. We did not find the target genes in most of the cases. So that means you have to go back and kind of overcome that challenge of identifying the target genes, thanks to all of you who really raised this point. And that's the first point I mentioned to you before, for the particular trait. For example, here in chickpea, you don't have that much, in pigeon pea or in green pea. And also you will not find much literature here because internationally, because these crops remain more important, 90% important for us, not as much because we consume and grow largest quantity in our country. So that way I'm giving you an example only of pulses, but about 10 to 12 crops. Again, I think I'll request Dr. Nilsing to kindly forward that paper, PDF to you. It's only about 3 to 4 pages. And out of these identified crops, 10 to 12, these are listed here. We developed a project. Dr. Vishwanathan would have talked to you about it, thanks to NASF and ICR. We developed a network project, and out of this network project, 2 events of rice are already in the field, undergoing field trials. One is Dr. Vishwanathan, that is using DST gene. Another is using CKX2 gene to increase the quantity of cytokinin in fluorescence, to increase more tiller number or more spike number in case of rice. This is by Dr. Satyendra in Hyderabad Rice Research Institute. And both have given here about 20 to 25 water use efficiency is increased. I said we should increase the efficiency of natural resources, water, land, or even fertilizers and natural kind of chemicals we use. But in here the yield per se has been increased 20 to 35% you compare under laboratory conditions. But I'm sure even if it is 10 to 12% under field conditions, it will be excellent, right? And they are both in 2 mega varieties, Remember, I mentioned to you before the elite variety in rice, not any other variety. Elite variety. This is MTU 1010, and this is, I think, BPT 5204, Samba Masuri. This is for the southern region, and of course, it is important nationally as well. So we are very proud of it. You must clap for them. These are the only two lines which have, and of course, Dr. Bish would have talked to you about mustard is coming up. Banana is coming up also. More crops are coming up. Okay, but then now I want to quickly go through the regulatory aspect, which is again a challenge. But I don't know whether it's been covered already, would have been covered. Why these crops are, GM crops we started with are regulated. Why GM crops are regulated from biosafety point of view? They are regulated because we allow a novel combination of genetic elements. Recombinant DNA technology, we use it. The promoter could be from any organism, coding sequence could be from another organism, terminator could be still from another organism. Okay, so these are actually speaking, novel recombinations taking place, and then we put it in the plant, which otherwise is not occurring naturally. Even in their natural genetic variation, you don't find that kind of a gene construct which you prepared, so therefore they need to undergo biosafety regulation. Okay, but here, fortunately, in SDN1 and SDN2, there is no such novel recombination of elements taking place. So therefore, what do you think? It should be regulated? It should not be regulated. Except SDN3, which is just like developing transgenic plants, that should be regulated. Because these are, I don't want to go into the details. 2017, only few countries declared if there's no foreign DNA present, like it is present in case of GM crops, if no foreign DNA present in the edited plants, they will be treated like non-GMOs. Right? 2017, only four countries are shown here in green. I think it's US, Canada, Brazil, Argentina and Chile. And then more countries increased number in 2020. India is still yellow, not green. We started discussion. And we fortunately had a lot of discussion and tried to, I prepared a video to request Government of India as well and other stakeholders that to help us out, at the level that we can have genome editing allowed, or without any biosafety regulation. Okay, and finally, this landmark decision, I call it, was taken, exemption of genome edited crop, particularly SDN1 and 2 from biosafety assessment, which was a very win-win situation for us. And that's why 2 of those rice varieties are already undergoing field trials. Okay, and these are some of the official orders. And this is the SOP. Fortunately, we were involved here in preparing. And here we don't talk about off-target, effects. We are not talking about off-target effects. What is important here is actually, though there are many other things required, evidence to confirm that genome edited plant is free from exogenous introduced DNA. They are anyway introduced, as you know, CRISPR reagents that construct we prepare, Cas9 and sgRNA. And there's a methodology you would have, again, you talk to breeder more about it, they will inform you. Okay, so, and then in 2022, you can see here, India also became green, though many other countries are having the same. And now the situation is very simple, that you apply directly to IBSC. Of course, we do IBSC, and this is going to play a major role. And we have got exemption under Rule 20. I'm sure Dr. Vibha Ahuja would have covered all this, but we struggled for it actually, and we came up to this level, and now it's very simple. And we made it simplified based on science. So that's what you should all understand. The science part of it is so important. And as a result of that, our Honorable Prime Minister, that time Honorable Agriculture Minister, Mr. Tomar, could announce, that we could get the first drought-resistant genome-edited rice out by 2026, and that is Dr. Vishwanathan's, using a DST mutant gene, which is already going. I said even it will be now another one, CKX2, for high yielding coming from Dr. Satyender's lab or Dr. Sundaram's lab, from Hyderabad. So we have now more projects. This is another opportunity for you all, friends. Do you know about this? That we have got 24 crops identified here, 15 crops identified here, and 500 crores rupees project has been allotted by Government of India. So a lot of opportunities. But unfortunately, you can see here, India is quite lagging behind compared to particularly US and China when talk of trying to develop genome editing, in case of plants. And out of 3 conditions, you can see 3 categories. SDN1 is mostly SDN1 knockout. And if you can do that, increase its efficiency further, identify target genes, there's a lot you can still achieve by SDN1. SDN2 is still a problem because of slow efficiency. But again, I'm sure Dr. Kutub Molla would have talked to you about prime editing or base editing, those are even better options than SDN2 for particular, targeting traits of interest. SDN3, of course, is like GM crops. It has to go again through regulation as far as at least India is concerned. So you can avoid that. Okay, and so therefore you got still enough to do. And finally, this is the future course of action for you all. These are the traits for the farmers. I don't want to go into the details, but important is functional genomics to target the target gene from the naturally genetic variation. You must have it, and then develop an efficient transformation protocol in elite crop cultivars is important. And then again, you can develop even a transformation protocol in a particular wild species which you want to convert into cultivable type, by retaining all those traits of climate resilience, etc. And then finally, can we make India as an international hub of you young crop genome editors? You are here for a purpose, friends. So not only, and you can take those challenges head on. We can all do, and we have to all do it. And I'm very happy you come from different institutions. By now you must have already been working into a particular WhatsApp group, and we have got several of those. I was told about 900 online. So what else we need in this world, in this country? We've got 1,000 experts already getting ready to take on genome editing, the challenges we're talking about it, and not forgetting the opportunity as much as we have to first take of our own country, and then of course finally rest of the world as well. You okay with me on this? You agree? I don't see that excitement. So with this, thank you very much. I don't know whether I have still time. Maybe I request organizers to give us 5 minutes for a general discussion on what I presented. You want to talk to me anything about it. And if there's not anything clear, you can stand up and ask me. Is it okay, Dr. Anil, for 5 minutes, 5 to 7 minutes? Please, open for discussion. And thank you very much. You can contact me with a specific issue. Okay, if I don't know, I'm very good at it in talking to Dr. Anil. He's my good friend. Many others in the country or internationally, or I can request Madam Jennifer Doudna to help us, and we can get the answer for you. So we can provide all that information, provided you are all ready to receive and make a difference. So thank you very much. Open for questions, please. Questions, please. Yes, please.
Sir, in your challenges, there is no screening points are there. So is it
screening for what?
Sir, for getting the edited cell from that populations after.
No. Once you have developed your T0, you have to validate. What kind of screening here? We don't do any kind of screening.
Means
validation of the edits.
Yes, sir. Is it not challenge for genome editing, sir?
No.
Okay, sir.
Validation, it could be, you have to do what sequencing. That's not a big challenge, as much you have a challenge of identification of the target gene itself.
Yes.
Followed by developing a functional protocol for delivery and regeneration of those edited lines. These are bigger challenges.
Okay, sir.
Once you have got an T0 edited line, I'm happy that I've got that line developed. Now I want to verify the edits. For this, there are various ways you can always do it.
Okay, sir.
That's not, I don't take it as a big challenge.
Yes, sir. Which are those ways, sir, through which
through
we
PCR you can do, or through even, sequencing. Mostly people do Sanger sequencing.
Yes.
PCR, of course, we recommend for confirming, after you've made a selfing or a crossing, that this is now foreign DNA free. So there in SOP, if you go, we have given protocols based on PCR alone.
Sir,
while sequencing can also be done there as well of the targeted region,
Sir,
but yeah.
If there is a bulk segregant, so for that also we can go on.
Bulk segregant kahan se aa gaya?
Means, sir,
Isme mein
bulk population is there lots of.
What kind of bulk population? I don't understand, sorry.
Means, sir, if you get lots of edited or non-edited population, through that we have to screen out or validate.
No, you do it by sequencing now. You do it by, first of all, you would have done it on selection mode anyway.
Okay.
On selection, you have been able to get everything positive on selection. Those only events you take forward.
Okay.
Yeah. But still, I think we can discuss it offline. No
Yes.
problems.
Okay, thank
we
you,
can
sir.
take that question there. Yeah. But otherwise, that should not be a major challenge. Yeah. Any other question, please? टाइम है, आप पूछ सकते हैं. हेलो, एक्सक्यूज मी, इसको जब तक निकाल दो, आप कर दो उसको. ये तो अच्छा, anyone who would like to ask a question online, please, online participants? Sorry, I can't see you actually speaking, therefore I tend to forget. Online friends, any question?
Yes, good morning, sir.
Good morning.
Yes, sir. I would like to know, sir, what is the off-target editing and why is it concerned, sir?
Actually, off-target editing happens because sometimes the HGRNA sequence which you design will be able to, get aligned with any other sequence near where it's matching nearby, close to it in sequencing, and Cas9 will quite possible make a cut there as well. Not at the precise site, there it will do, but it might even cut on other, sites on the genome wherein there will be some kind of a matching of that HGRNA sequence, though you have taken it precisely, but still that happens. And also Cas9, is not as precise as we want it to be, though there are certain variants, people are working on it. They're doing engineering to, making it more precise catalytically, though efforts are being done. But yes, off targets do occur. But the point is, once we know off targets occurring, we don't worry too much about in case of plants, as long as the phenotype, the kind of a trait of interest expression is concerned under field conditions, we only select those which are otherwise the best for us agronomically speaking. So we don't want to give too much emphasis on off targets because we know that we have got lines already created in the world, using other methods like irradiation, chemical mutagenesis, where people have studied that off targets, will be at so many places we don't even know, we never analyze those. But what we have only selected and screened thousands of those mutant lines. If we get the best particular trait we wanted to, we selected it, and there are more than 3,000 varieties, already in the market. So we should not give too much of emphasis. Therefore, we have avoided it, actually speaking technically, in our SOPs. So kindly go through our standard operating procedure, which is available on DBT website. And I will request Dr. Anil Kumar Singh to share with you all, including that Nature paper, current science paper, to all of you. Thank you.
Okay, sir.
Yeah.
I have also one question, sir. Can you give answer, sir?
I think you can email us, or you can please write in your chat box, because we would like to give time to others as well. One question, only one person with a large crowd we have. Thank you.
Okay, thank you so much.
You can email me. You can email me. Thank you. I'm very pleased. Where are you from?
Yes, sir. I'm sir from Maharashtra, sir.
Okay. Thank you.
Yes, sir. Yes.
Any other question from online participants, please? Okay. In that case, friends, thank you very much, and thanks to the organizers. I wish I could spend more time, but I will come again. Thank you.
Thank you so much, sir, for spending your valuable time this morning and presenting your talk. Thank you, sir. Yes.
Recording stopped.